Thank you very much for the introduction, and good afternoon to everybody. Since this is a post-lunch presentation, I'll give my best to make it as digestible as possible. So the outline of my talk is presented on slide number 3, after a very brief introduction into the product, to the process, and how we actually made the submission. We will actually touch on the following topics. The first one being, as already mentioned this morning, how to assess criticality of process parameters, how to deal with DOEs, in finally defining a design space. Then we will move forward to the control strategy and see how models, multivariate models as content uniformity NIR or even multivariate statistical process control models were utilized as part of the control strategy. And finally, we will also touch on some daily life aspects when QBD is actually lived in the production side and when it comes to problems or challenges related to change management, deviations, and also preparation for an inspection. So to start with, the product we are dealing with is actually a dispersible tablet. We are producing this in 3 different dosage strengths. The drug load is 30%, and the indication is actually a chelation therapy for the management of chronic iron overload. Below, you can actually see an overview of the unit operations, both on the API, but also on the drug product side. For the down processing on the API, we have actually a sequence of charcoal treatment, crystallization, drying, and milling before we are proceeding to the secondary manufacturing, which is a classical solid dosage form process comprising the unit operations of high shear wet granulation, drying, blending, and compression. All these unit operations were considered in the development of the QBD submission. The product is actually, as you can see it from the title, a legacy product. So initially, the first filing was made in 2005. Then in 2006, this product was identified as being the first pilot project for QBD within Novartis, mastering a significant amount of activation energy, and finally submitting the dossier in 2008 for the drug substance and for the drug product in 2009. As previously mentioned, we were covering both, so the last steps of the API production, as well as the complete chain of unit operation for the drug product side. This was also an excellent opportunity to introduce new concepts for the control strategy, such as real-time release testing based on design space, based on PAT, on empirical models developed also in the respect of blend uniformity. And also finally, we also made use of multivariate statistical process control, which I explain later onwards, where we made use of data which are anyway being recorded. Pre-approval inspection took place at our Swiss sites for both for API and the drug product. In the next slide, you actually can see the roadmap of our risk assessment, how we ended up in our design space, how it was actually verified, commencing actually with a basic risk assessment, which is called QBD1 document, where we are capturing all the available experience from the previous submission, as well as we are speaking of a legacy product, also from already available process knowledge. Here we could filter already which process parameter could potentially be deemed to be critical, and all these potential process parameters were then actually included in a sequence of different lab investigation by means of DOE at lab and pilot scale. Then the second risk assessment was conducted after conclusion of the development work. Here in this document, which is called QBD2, the risk assessment was re-evaluated, and also the control strategy was defined with the design space, and then we moved forward to the scale up, to the full-scale confirmation at manufacturing scale, where the design space was verified, concluding the risk assessment in the last document, which is called QBD3, where we actually did the final ratings for all the different process parameters, verifying the criticality of the process parameters which were identified during development. For the FMEA, for the risk assessment, we used the following quite common metrics to better structurize the process parameters. All the different process parameters were depicted on fishbone diagrams. We used a 5-level scale in order to assess the risk priority number, ranging then from 1 to maximal 125. As a threshold, we agreed to set this as 16, which is the average of all the factors being at 2.5. And that means that all values above 16, process parameters with values above 16 was considered to be carried over in the next phase and have to be investigated by means of DOE. To mention an additional point is also that severity was also deemed as an additional threshold as a requirement for including the parameters in the DOE, since the criticality is solely dependent on risk and the high detectability does not actually mitigate the criticality. So to become a bit more less abstract, we are now dealing with a very concrete, tangible examples, and we are now focusing on one unit operation. We picked up the granulation. We have various process parameters which potentially have impact on the CQAs, like dissolution or assay. As you can see in the FMEA table, we had a glance, for instance, on the spray rate and the amount of water which was actually used during the granulation, also the mixer speed. This is the assessment which is reflected in the so-called QBD1 document prior to the execution of any kind of experiments. So here the potential impacts on the output quality are quite high. So here this is resulting in also high RPN numbers, with the consequence that these parameters should be also considered and evaluated during lab scale developments. How this was actually arranged and managed can be seen on the next slide, where you can see the setup of a DOE, including exactly these parameters which were filtered out of the risk assessment. So for the granulation step, in total, we had 5 process parameters. And with the fractional factorial designs, we could actually explore the criticality of each of these different process parameters, carrying out 20 experiments, 60 of them part of the factorial experimental design, and 4 experiments on target replicates conditions. The output of this is then seen on the next slide, where you can clearly see out of the 5 process parameters, only 1 actually proven to be critical. The one which was critical is the water amount, which had a significant indication and a high p-value. Coming up in the data analysis of the DOE data. And this is now reflected in the so-called QBD2 document, where you can see on one hand, the amount of water still remains quite critical from an impact point of view. So we still have a 3, whereas the mixer speed had no obvious impact on the final quality. And therefore, it was actually downgraded from 4 to 1 and was no longer pursued in the subsequent development work. So that means that of all these experiments, what was the conclusion? That we should consider the amount of water as a critical process parameters in our full-scale investigation. So how did this work for all of the process parameters? As I already mentioned, we were dealing with 4 unit operations. So for each of the unit operation, we carried out such a kind of a DOE to assess the criticality of process parameters. In addition to that, we also evaluated the risk concerning the material attributes, and for that, separate DOEs were executed, varying, for instance, excipients coming from different vendors, also varying the particle size distribution of the product, and also quality characteristics of the drug substance. Out of all these evaluations, then we actually came to the conclusion what of these process parameters and critical attributes were deemed to be critical, and these were separately also investigated in the so-called grande finale DOE to additionally also assess the interaction between the critical process parameters. And as a final step, then on full scale, we also did a DOE to confirm the criticality and the ultimate design space. As an overview, you can actually see what is the outcome of this investigation. Among all of the different potential process parameters, it turned out that only the particle size distribution of 2 excipients, in addition to the water amount, was actually critical. What we then concluded is that the particle size distribution is not scale dependent, therefore it was not further considered in the full-scale confirmation. And to increase the variability in the full-scale confirmation DOE, we also added some non-critical process parameter. So in the end, we assessed our design space at full scale with 1 critical and 2 non-critical process parameters. leading into 8 process parameters, and then ultimately in a design of experiments with 8 batches to be produced. And with that, actually, then this was the last step in the confirmation of the design space. And with that, I want to hand over to Lama, who should give them the regulatory point of view.
Thank you, Lorenz. So I'm going to present our observations on this first discussion topic. So from our point of view, we have here in this case study a satisfactory way to perform the risk analysis. We define analysis of the process using fishbone diagrams and a clear risk assessment methodology using FMEA metrics. But most of all, it's important to highlight that here, risk assessment was driven by severity. So even if a high detectability was in place, this was not used to mitigate criticality, as Lorenz pointed out previously. Regarding the outcome of the design of experiments, and in particular the statistical evaluation, we were content in this case study with Pareto charts only. We did not further focus on the modeling because the design space limits were not extreme. And although the design space was the surrogate for dissolution testing at release, we took into consideration that the drug product was a dispersible tablet, so dissolution was considered as less critical. But in principle, for design of experiments that are used to establish a design space, we need more statistical evaluation in order to confirm the validity of the model. So we would ask for the results of the model diagnostic tools, such as a goodness of fit, goodness of prediction, ANOVA p-values, etc. Another particularity of this application is that full-scale design of experiments were executed. And here we would like to note that full-scale design of experiments are not mandatory. In this case, it was probably easy to perform design of experiments at commercial scale because the product was a legacy one. So regarding the recommendations, we have chosen to address the level of details that is needed in the application for the review of the risk assessment and of the design of experiments. So as you can figure out, this will depend on the purpose and the use of the studies. So if a design space is claimed, we would appreciate to have a comprehensive risk assessment because this will help us understand the rationale used by the applicant for the selection of variables that require further studies. So we would like to have the individual scores and the threshold with a rationale for it. The risk assessment can be presented in the form of a risk matrix, but the presentation provided in this application is also very appreciated. So as Lorenz showed previously, this was given in the form of a table listing all the process parameters, the variables, with their respective scores, a brief rationale for the scores, and the final risk priority number. Regarding the level of details for the design of experiments, here again, it will depend on the purpose. So if the design of experiments are performed for screening purposes only, then a summary description of the experiments and their outcome is sufficient. But when a design space is established, we would appreciate to have more details. And as you can see on this slide, we would need to have the type of experimental design because this will allow us to know if the resolution of the design was sufficient to establish not only the main effects, but also the interactions. We would like to have tables summarizing the inputs, the ranges, and the results achieved for each experimental run, not forgetting, of course, the batch size. This will help us understand the variability introduced in the design of experiments, and also it will allow us to check the distance of the response to the specification. If the design of experiments have been performed at lab scale or pilot scale, then we need to have a discussion of the scale-independent factors because this will have a consequence on the extent of design space verification at commercial scale. We've already addressed the statistical evaluation, so we need the statistical significance of the parameters studied with the interpretation the applicant makes of them. And finally, we would appreciate to have a summary of the parameters that were kept constant during the design of experiments. This is in order to have a complete information and in order to be able to verify that those parameters were set at the same levels as described in the process description. Sorry, that's not. So to continue with the recommendations, we already addressed the first point. So we said that the use of commercial scale batches for design of experiments is not mandatory. So the design space can be developed at lab or pilot scale, but in that case, we need to have in the application a protocol for design space verification at commercial scale, and this has already been addressed in the previous presentation. It's really important for us to have a clear and transparent control strategy. For example, we need to know if really a design space is claimed or we have only parts that have been investigated for robustness purposes, for instance. This is essential because it has a consequence on the requirements and the data scrutiny by authorities. Although a design space would normally include only the critical process parameters and the critical quality attributes, from our point of view, this doesn't mean that the non-critical attributes and the non-critical process parameters can be omitted from the design space description. Actually, irrespective of the development approach, we need to have a process description that is sufficiently detailed and that will include the relevant process parameters. And to finish with the recommendations, we think that the description of the design space really need to be presented in the CTD sections giving the process description, because this is part of the regulatory commitments, and also because it will facilitate the review by the assessor. So by looking at the sections giving the process description, the assessor can immediately understand the control strategy and the extent of flexibility claimed by the applicant, and therefore the amount of data that are expected in the application. This slide shows the way the process description was presented in this case study. So from our point of view, it's a satisfactory way to present things. So section P33 of the application included tables giving all the target settings, one table giving all the target settings, another table giving the critical quality attributes and critical process parameters that defined the design space with their ranges, and a final table listing the critical attributes and process parameters, the quality attributes and process parameters that are not part of the design space with their proven acceptable ranges. And so I will hand the floor to Lorenz again for discussion topic 2.
So we are now moving to the second topic, which is dealing with the implementation of multivariate models in supporting the QBD control strategy. For that, we have examples coming from content uniformity and blend uniformity, but we also touch on the applicability of multivariate statistical process controls. As an introduction, there's a categorization of models depending on their impact to the release mechanism. So we are speaking of high impact models, which are the sole indicator for the quality and release. Examples for that would be the NIR for content uniformity, which would be a one-to-one surrogate of the already established HPLC model. Medium impact models could be considered, for instance, the NIR for the blend uniformity, which is, so to speak, an in-process control in order to control our blending process, as well as the multivariate statistical process control models for granulation or for drying, which is assuring that we are still operating in our expected normal operating conditions. Lower impact models, as we interpret, could be actually main effect UEEs, which are carried out at the stage of development. So in the following slide, you can actually see the control strategy of this process, which is only reflecting the drug product part. So we have 4 unit operation: granulation, drying, blending, and compression. For the blending and compression, we made use of the so-called hard PAT instruments. So we actually used PAT NIR spectroscopy in order to assess the blend uniformity, in order to assess the individual potency of tablets, where we could actually increase the sample size. But we are of the opinion that we do not have to implement PAT for every unit operation. We should do this on a risk base, also driven also by business arguments. And therefore, for the granulation and for the drying, the decision was actually to utilize the already available process parameters as stirrer speeds, as temperatures and pressures, and combine and condense them in form of an MSPC model. So for each of the unit operation, we had a dedicated model, which then served actually in increasing the detectability for our real-time release approach, which is then reflected in the table. So for content uniformity, ID, and assay, we used actually a surrogate, a hard PAT method, which you can actually see on the slides. For instance, this MPA content uniformity instruments. For dissolution and degradation, we made use of so-called soft PAT, soft sensors. And here actually the MSPC models in combination with the design space served actually as our real-time release testing procedure. Then the question is also how to validate these kinds of models, especially when we are talking of high impact models. For the validation of such kind of empirical models, you have to actually proceed through different stages, collecting different types of data. So you're starting actually with collecting, measuring a so-called calibration set, where you have actually an extreme variation of the concentration. Then you are able to build a model, which is then confirmed by an independent data set, which is already available at time of development. And with these two sets, you're actually able to finalize your model. Then after the model is finalized, you fix it, and then you are proceeding to the so-called external validation, which is a punctual assessment of a limited number of batches, for instance 3, where you actually to prove that your model is coping with data, with batches which were not available at the time of developing. So you have to deal with the unexpected future. Then the peculiarity of NIR and empirical models is also that you also insert a so-called parallel testing phase, where you still do not actually implement the method for the full release. So you still run your HPLC method in parallel. And the reason, the objectives for that is actually in collecting a statistically significant higher amount of batch data to have a large body of data where you also have statistical confidence that your model will work and will also cope with the expected variability of your future production. So the three first rectangulars, the calibration set, test set, and validation set is actually the classical method validation as you do it also for HPLC. This should also be subject to submission. The parallel testing is actually a volunteer activity to increase the confidence in the method and should only be subject to an inspection. Then to give you a better flavor about the journey, how to develop and to validate and to implement such kind of a high impact model, you're starting from a risk assessment, you have to come up with a calibration design, you're doing a lot of feasibility studies on target batches, then you are proceeding to the data collection phase, you're producing calibration tablets ranging, having a wider range of your labeled claim. Then you're defining your acquisition parameters, and then you are proceeding onwards to the model generation, to the modeling activities, where you then in the end finalize the model, and then as a last step, you're doing the external validation with a functional assessment. Sometimes when the model is developed in development at the different site, you have to transfer the model as part of a classical GMP transfer. And then after parallel testing, you're implementing the model for release, and then you have to do your maintenance as part of your lifecycle management. When it comes to robustness, you also have to address potential factors which can have actually an influence on your predictive performance. This should also be anticipated in form of a risk assessment. For instance, for this content uniformity NIR method, we also explored the influence of different excipients. We also were able actually to conduct and to assist all the DOE activities where we had already automatically varying process conditions. We also had to look on the hardness and the influence of Bosman operators and how we are presenting the tablets to the sensor. How can you check this? Actually, there are two ways. Either you are incorporating this variability by design into the calibration, or you are confirming this afterwards by testing. Then when it comes to the validation and having a specific look on the validation criteria, we are distinguishing between internal validation and external validation. For the internal validation, we are using and applying statistical acceptance criteria because we have actually quite a large body of data. So here we are investigating a population of different batches and looking on the average performance. In contrast to that, for the external validation, we are looking at 3 individual batches and assessing these individual batches by individual acceptance criteria. Therefore, the bias, which is actually the difference between the reference and the NR, is the internal validation in the statistical assessment much lower than for the individual assessment, where due to the distribution, also this difference can be quite high. Other points to be considered as part of validation are linearity, specificity, and precision. Linearity is normally covered by the accuracy across the range, also by the distribution of the residuals. Specificity, here we compared the placebo tablets to the verum tablets and also could show that we had a specific peak for the API, which is actually a luxury for NIR, and for reproducibility, what was actually investigated. And closer seen was actually the influence of operators doing the measurements. After the validation, then we decided to run the methods in parallel for at least 15 batches prior to the final implementation as part of our release procedures. With that, I want to hand over again to Lama.
Thank you. So the first 2 observations are linked to the fact that we were assessing a legacy product. So at the time of submission of the QBD application, the state of knowledge was quite substantial. So we accepted that a weight correction was not needed in this case because we had sufficient evidence that weight was tightly controlled based on the long experience and the process capability. We also accepted a reduced range for the near content uniformity, although in generally the recommended range is 70–130% in order to cover the requirements of the Pharmacopoeia European monographs for content uniformity. In this case, we accepted a reduced range because we had data on about 200 commercial batches showing a very tight and uniform distribution of the drug substance. And also we took into consideration that this was a high dosage form and that a control by NIRS was introduced upstream for the blend step, as we will see later on. Going now to the third bullet, the NIR guidance requests that the external validation is performed on an independent set. In this case study, accuracy across range was demonstrated on tablets coming from the same batches used for calibration. So accuracy across range was demonstrated not on independent batches, but on independent tablets. And we considered this acceptable. Just for clarity purposes, we told the applicant that in this case, the model for content uniformity by NIR could not be considered as a real PAT application since it was performed only on 30 tablets that were analyzed offline. And also we asked the applicant to clarify in which circumstances he could go back to the old reference method, the HPLC method. And so it was clearly stated in the application that this could be done in case of failure of the NIR apparatus, investigation purposes, and also even if a NIR result was obtained outside the validated range. And to conclude with the observations, we did not request any NIR raw data because we do not perform a recalculation of the model. We just we considered that typical NIR spectra were sufficient in the application. So as regards the learnings, since the time of assessment of this application, the NIR guidance has been updated, and now it has introduced the concept of scope that will facilitate continuous improvement and that will show how changes to the NIRS procedure can be implemented from a regulatory perspective. So the elements of the scope are detailed in the guideline. I won't list them here, but just for illustrative purposes, in this case study, the scope was not clearly identified as requested in the guideline, and some elements were missing. So we would ask for this information if the application is submitted today. Regarding the parallel testing, in this case study, the number of batches planned for parallel testing was 15, which is satisfactory. But actually, there is no standard regulatory requirement for parallel testing for the number of batches to be included in this step. So we propose this as a topic for further discussion, and we think that there might be room for further guidance on this topic in the future. For the moment, the assessment is done on a case-by-case basis based on a justified protocol submitted in the application. I think this ends topic 2, and I don't. Yeah.
We're coming now to the second example, which in this case is actually a medium impact model. And we're dealing now with assessing the blend uniformity of our bins by applying NIR spectroscopy, which was actually the first time we were introducing this technology into our manufacturing area. The goal was actually to come up with a quantitative model. This model was validated and developed at our lab scale development center in the US, and subsequently then transferred from the US to Switzerland. On this picture, you can actually see the dimensions between the spectrometer and the bin blender on lab scale. And on the following slide, you see exactly the same instrument hooked on a 750 liter blender, and you actually see that the ratio of the dimension has dramatically changed. So the objective of the method was to have an online API prediction. It was a quantitative model. We were using moving block standard deviation of the API to also address the standard deviation. And as a last step, we wanted also to make use of this method to control the blending process, meaning that we were relying our blending process no longer on the fixed time, but on the NIR signal. For that, we had 2 criteria. So the API concentration had to stay between 19 and 110%, and the standard deviation derived from the moving block standard deviation should be also below a certain threshold, which was determined on empirical data. When those 2 parameters were actually pertained for a certain period of time, then the blender was stopped automatically. The effect of that and the comparison with the classical approach can be seen on the next slide. In blue, you can see the classical approach based on a fixed time, opposed to the new approach where we are now stopping our blending process based on our NIR signal and based on the fulfillment of the 2 criteria of the API concentration. And as you can easily see from this slide now, the blending time has significantly decreased. What has to be taken into account, into consideration when developing, validating, and transferring such kind of a method? First of all, we had to build external samples representing a wider range of the API concentration from 70 to 130%. It was actually an offline model where we actually did this external calibration with static samples. Then we showed and demonstrated the performance of these methods in development in the lab scale on DOE batches, where we could vary quite a lot of different blending parameters like the RPM, the fill level, also the raw materials which went into the blending step to show and to prove that our model is still predicting accurately. Then after the validation, we had to transfer the method which was developed at lab scale to full scale and to prove comparability between the different scales and that the method still is actually sufficient from a predictive performance point of view. And then at the very last stage, we also qualified and validated the process control. That means we actually stopped a batch depending on the already mentioned criteria from the NIR, and then had a look on the batch whether it's actually meeting the blend uniformity requirements. The tricky thing hereby is actually what are you taking as a reference?
Regarding the observations, during the preparation of this presentation, we had a discussion whether blend uniformity by NIR should be considered as a high impact or a medium impact model. So as Lorenz pointed out previously, it is perhaps somewhere in between, but we as assessors, we were of the opinion that it should rather be considered as a medium impact model in this case, because it was not used for release testing and also taking into account the high drug load in the blend. During the discussion, we also agreed that there were still question marks about the requirements, the validation requirements for NIR methods when they are used as PAT tools for blend uniformity. So regarding quantitative methods, we were of the opinion that the requirements of the NIR guideline can be applicable, with a question, however, about the sampling for the reference method. We thought that because of the inherent variability of thief sampling, it is perhaps preferable to replace thief sampling by final testing on the compressed tablets. Regarding qualitative methods, the requirements of the NIR guideline might not be applicable as such, so there is perhaps room for some kind of guidance on this topic in the future. And that's all. I will leave the floor to Lorenz again.
Last but not least, I also want to mention the application of the so-called multivariate statistical process control models. These models are seen to be soft models, so you don't need to integrate a sensor into your manufacturing equipment. You're making use of data which are already being sent to your SCADA system. In that way, you're exploiting already available data and harvesting low-hanging fruits. To visualize and to explain this a bit better, you see on the left upper corner actually the process parameters coming from the SCADA system, from our granulation process. So you have the power consumption, the torque, the temperature, the amount of water which is actually fed into the granulation vessels. And as you can see, all this data is highly correlated to each other. There's actually a certain principle behind this, but only an extremely experienced Swiss operator could actually tell us whether this is actually a good batch or whether we're actually facing a problem. To overcome these problems, we condense this information into a latent variables by means of PCA and are now following not the original process variables. We are now monitoring this latent variable as a function of time and putting this into statistical context, enabling our process experts and operators to make a very fast assessment whether we are in normal operating conditions or whether there is a tendency that we are departing from these conditions and have an excursion or a start of a problem.
So just one remark on the MSPC for granulation and drying. We considered it as a low impact model in this case because the manufacturing process was already validated since the product was a legacy one and the design space was verified at commercial scale. So the MSPC model here was rather considered as part of a continuous process verification that supported continual improvement. And as a consequence of this classification, we considered that the validation data of the model need not be submitted in the application, but they should be available for review during routine GMP inspections. And so to conclude with the recommendations on the models in the control strategy, we would like to stress that the level of details will be correlated to the impact of the model. And this is why we need a clear and transparent control strategy in the application. So for example, we need to know if the design space is a basis for real-time release testing or not. If a PAT tool is described, is it used for release or is it just used for in-process monitoring? If there is a monitoring of the process, is it used for continuous improvement or is it really an alternative approach to validation? So depending on the answers to these questions, the impact of the model will not be the same and the data requirements will not be the same also. And so I leave the floor to Florence again.
We are now coming to the last topic, which is more dealing with the daily aspect of QBD in real production life. So the product is now being produced since 3 years, and we also made a couple of experience how QBD is then handled in daily manufacturing. And there are different types of issues which might occur. For instance, how to handle deviation, OOS and OE results. For instance, what happens if an NIR measurement indicates that you are out of your specification. And secondly, how do you handle excursion of the design space, which was already discussed in the morning. Then also changes is also a very important topic, since it's actually in the nature of these kinds of empirical models. Any type of slight changes in your manufacturing process might have an impact into the predictive performance of your NIR model. So there might be certain circumstances where you're actually forced to change your NIR models. And last but not least, we will also think about the general expectation, how to prepare for a QBD inspection, for a pre-approval inspection before the implementation, but also in the context of a general GMP inspection, which is more dealing with the lifecycle management aspects. So the first topic would be actually what happens if our continuous uniformity assay NIR models would actually tell us that our tablet has only an assay value of 92%. So then we could not actually automatically refer back to our HPLC method. We have actually to conduct a thorough and structured investigation to exclude different sources of errors. The first one would be actually a standard investigation in the lab to exclude, first of all, a lab handling error. We have actually to repeat exactly the same measurement on the exact tablets in order to verify that the result was really out of the specification. If this was just a handling error, that the operator placed the tablet in the wrong position, then we could actually invalidate this initial OOS results by 6 consecutive new measurements on the same tablets. If we exclude a trivial lab handling error, then the second source of the error could also a sampling error. The tablets could have been damaged or something have been, there must be a contamination to the tablets. Therefore, we would actually sample an additional set of tablets, would repeat the NIR measurements to see whether the OOS results would be confirmed or whether they will be in spec. In the case, if this is still confirmed, the OOS, then we would actually proceed to an intensive investigation. We would review all the process parameters, the batch record, and as part of that, we would also instruct an extensive testing on the IPC, but also on the finished product side, having all the information together that we can actually make a final decision whether we have a false negative result from the NIR or whether we have a real deviation, a real OOS results for this specific batch. In this case, we would reject the batch. If we would actually come to the conclusion that the NIR method is insufficient, that would actually trigger a kappa in updating the NIR method. So this would be the procedures for a hard PAT measurement failure error. But what happens if, coming back to the granulation example, if the operator by mistake is actually feeding too much water in the granulation vessel, that we would slightly be out of the design space. So we would not meet the 34% criteria. We would be at 35%. What would be the subsequent actions? In this case, we would actually evaluate the extent of deviation. Is it 35%? Is it 90%? We would conduct a risk assessment to evaluate the potential impact on quality. We would evaluate whether process deviation can be handled by subsequent steps, for instance, in the drying, that this could be compensated. We would do also define a scope of additional testing, IPC, LOD, PSD, and then we would also refer to the full end product testing needs to be done to make a full judgment and an assessment of this, the status of this respective batch. And with that, I want to hand over to my colleague, Jürgen Melitz.
Okay, thank you, Lorenz. So at this point, I would like to give some input on the GMP topics from the viewpoint of a GMP inspector. Okay, yeah. The handling of a design space deviation might be, of course, a very interesting topic throughout the GMP inspection. And from my point of view, it is very important that, first of all, there is a strategy defined how to handle such a deviation. And there should be a very clear documentation of the complete deviation. And it should be very transparent too. And there should be on every step of this deviation handling, clear risk communication to enable the decision takers, to take a decision about the batch. So how to handle it? There should be an immediate action. There should be a risk assessment and maybe additional sampling to get an idea whether it might be useful to go on. If the output of this risk assessment is it is a severe deviation, then there's a no-go at this point, and the batch might be discharged. Otherwise, if the decision is taken to go on with manufacturing, maybe with additional sampling if necessary, then the question arises how to handle this batch. Is how to come to a release decision. And from my point of view, it could be pragmatically in the way it is written here on the slide, and having in mind scientific data from development and maybe the process knowledge that might be available at site. So there should be the decision to make a full quality control and testing, no real-time release testing, and a thorough batch record review combined with risk assessment. And the outcome could be, from my point of view, that the batch might be released if all those data, the additional data and the full-end testing and the risk assessment leads to the decision that the OOS of the design space is invalidated. So NIR is quite often used for PAT applications, and so we take this example for another interesting point: change management, post-approval change management. Sorry. So, of course, for an NIR method, there should be a scope defined, and this procedure should include, for example, the instrument, so from which supplier, and the substance to be tested, and the method characteristics, and details about the model. But of course, in daily life, it might occur that the scope is changing. It might be useful to have, there might be the need to have a new NIR spectrometer, or the model has to be updated due to a supplier change, for example. And for those post-approval requirements for NIR procedures, you can get some help in the note for guidance of NIR. In the latest revision, there's said that changes outside the approved scope are subject to variation application. And normally, this might be a type 1B variation, but in case the company provides a change management protocol, it might be only a type 1A variation. So this change management protocol should, might be a way to have a faster and predictable implementation of post-approval changes, and it should, for sure, as a crucial point, include strategy and test procedure, and this should be agreed with the regulatory authorities. Examples for this are on the slide, for example, an update of a spectral library for identification, and so on. So because time is running a little bit, I will just go to the next slide. And I was involved in the pre-approval inspection regarding this case study. And from my point of view, it is very helpful to have, for the preparation of the inspection, a very good exchange with the assessor, and after the inspection too, to get an idea about the deficiencies and in which ways they have to, they could be mitigated. And so from my point of view, the interface with the assessors is very important, and it would be, from my point of view, ideal to have them, to have one of them with us on the GMP inspection when we perform the GMP inspection. So from my point of view, it is a crucial point of course, as a basic for all this, to have a complete and sufficient qualification of the PIT systems, starting at the user requirement specifications and going through the complete qualification process. And a very special and important topic is from my experience I got from inspections that the qualification of the automation and IT infrastructure, the computerized system validation is very important, especially the transfer of the data from the probes to the system and the interfaces between the probes and the data storage and data storage systems has to be proved. This should be definitely a well-structured part of the qualification. Another point we would like to stress on during such an inspection is whether there is a periodic requalification implemented and which topics are included in this. And of course, some other points you might know from routine inspections too, like implementation of a maintenance procedure and the system SOPs, how to handle change control and out of specification results. A specific topic of interest from my point of view is to have a look on the training records of the operators which are involved in this specific operations, for example, handling the NIR instruments or the evaluation of the data. And for general GMP inspection, and now I'm near the end, sorry for talking so much. At the end, from my point of view, a very important topic is for the routine GMP inspection, and this might be really tricky and not so easy for a GMP inspector on site to get an idea about the method performance of this PAT and quality by design elements. So a chance to evaluate this might be to have a look on the product quality reviews and the results of the continuous process verification, and together with a look on the deviations and changes, we might be able to get an idea whether the performance is good or not. And another very important point is about the batch release procedures, that it is clearly defined at which point, it was mentioned before from Lama, at which point it is allowed to switch from the real-time release testing to the conventional testing, and to have a very, very clear strategy how these batch releases are done. And as a result of all this, there should be a system, a procedure to identify, evaluate, and implement points of continuous improvement. So thank you so much for your attention.
So I hope we managed to keep you awake and would like to address your questions. Yes?
Matt Bopkin, GlaxoSmithKline. Thanks for a really interesting talk. I think that especially the stuff around impact of models and the levels of detail that we're expected to see is going to be really significant if we can get that uniform agreement on that, because a lot of the questions that certainly we find that come back on submissions are related to that level of detail. So if we can get ourselves to a position where everyone is comfortable with what's expected, I think that'll reduce greatly the level of questions we have. My point I wanted to raise, kind of two connected points, was you made the point very clearly, which I think is a very important point, that the level of detail in the submission related to a model should be related or correlated to the impact of that model, and I think that's a really good point, and I'm glad to have heard it made. I would like to challenge one of the points that was on one of the slides, that a design space model would necessarily be a high impact model, and I think actually that was on a slide, but I don't think necessarily it's something that is always going to be the case, because, for example, if your design space is only a part of your control strategy and you have end product testing, clearly in those situations you wouldn't think that a design space model would be high impact. And my second and related point was that there was a slide, I think slide 13 or 14, where it talked about what level of detail would be expected to be in the dossier to talk about the model that defined the design space, and it talked about a lot of the statistics wanting to be present there. Sometimes when you define a simple design space, you often identify your critical process parameters and the interactions with a more broader screening design, and the final design space defining DOE could be across much narrower ranges where you don't see interactions and you don't see critical effects. And in those cases, I don't believe that the sort of statistics that were listed out would always be relevant, although clearly outcomes from design runs would be. So it's quite a long, complicated question, but I guess what I wanted to talk about and to get comment back on was, you know, is it one size fits all for a design space, or do we have to think more deeply about what that design space is used for and what type of model supports it?
Thank you very much for this excellent feedback. Speaking for the design space.
I can try to give a response to the first part of the question. So the design space will be considered as a high impact model only if it is used as a surrogate for release testing. And this was the case here because it was used as a surrogate for dissolution release. In the other cases, I think that if it's not used for real-time release testing, then it should be considered as a medium impact model only.
You mentioned just that point around there was a list of what expectations would be in terms of the statistics that underpin any design space model. Can you comment on that point I made about sometimes those aren't really meaningful if you've actually got in a robustness type DOE where you don't see effects across the range that you've studied?
If I understand well your question, you mean when the response, in fact, is very far from the specification and there is no real impact on the variability. Yeah. Yes, I think that in this case, I agree that we can be less regarding on the statistical evaluation and the robustness of the design of experiments. Yeah.
To some extent, if I understand well, you are in the situation where you have non-critical parameter. Am I correct? You demonstrate through the DOE that you don't have any critical parameter and the ranges are extremely narrow.
No, actually, you may indeed have critical parameters, but the boundaries where you set your design space, you actually don't see any impact, and therefore the final DOE that defines your design space won't give you meaningful statistics about your product. And it's actually the experimentation itself which defines where the boundaries will be.
So my question is, when you have very narrow ranges, what is the added value of claiming a design space? This will be added to the discussion we will have tomorrow.
Yeah, it's a good point.
Yeah.
Just intervention. What you've seen in the presentation is like what you would normally expect for a design space which is coupled by some sort of release testing, so it's going to be medium impact, and it assumes that there are interactions between parameters. First of all, because you can have a design space where we see no interaction, so then it doesn't make sense to look into models and all this statistical fitting. So again, we are trying to cover 80%, you know, the Pareto approach as well. Even here, we're trying to cover 80% of what we normally see in a submission, and the rest would be on a case by case. But then it would be very useful if in your dossiers you would explain, we don't go further for this reason. And that would help the assessor. So yes, there are a lot of cases where maybe additional statistical analysis will not be needed, or maybe, again, I would go to, we've seen also some funny examples where you see a design space with very narrow ranges and you wonder why, but this goes to the presentation that Graham said at the beginning, do you always need to file a design space? But answering your question, yes, there are cases where that level of statistical detail might not be needed, but what we would expect to see is an explanation why, to facilitate the assessment.
Yeah, over there.
Yeah, thank you. I want to come back to the first point that was raised, because I agree with with what was said, but I think there's an important nuance to that. It's not always either design space or end product testing. At least in the biologicals field where I come from, we often see that the final quality of the product is determined by both the end product testing and the process, and especially for impurity removal, I think this thinking may be translated to the small molecules, and so I think this is something that should be taken into account.
Yeah, Dora, please.
Lorenz, with the danger of eating on my time, I'm just going to ask a question. When you do the content, are you using a tandem system, or are you taking the tablets and you go to the lab and you analyze them?
At the time where we introduced the technology, the tandem was actually not in a mature state to use it. So for subsequent project, we will use actually an online handling of tablets, but for this, actually, the tablets were transferred manually to the probe.
Okay, good. Thanks.
Last question.
Lorenz, that was a superb presentation. You covered such ground in great detail. I could ask you lots of questions, but I'll restrict myself to one, and possibly to Lama as the regulatory voice. I was particularly interested, and this is a non-technical question, I apologize after your wonderful presentation, but on slide 15, you have a point that I'd like to just explore a little for the audience. It's you need a clear and transparent control strategy. That's been coming through across the day. And then there's a very fascinating comment made that says if a design space is claimed, or if PARs have been investigated only for robustness purposes. That's a fascinating sentence, Lama. What would be the difference for the applicant if they went design space or PARs in your perspective?
I think that this will have an impact on the amount of data we need on the design of experiments. For example, if you are claiming a design space, then we really need to be sure that the design of experiments performed are of sufficient resolution and that they are able to detect interactions. In case of PARs, univariate experiments could be acceptable, or maybe experiments with a less robust resolution.
I think this is really important for the whole workshop, really, to try and get these nuances explored.
I think, though, we discussed this morning when we had the first presentation, and we discussed whether or not, essentially when there was the question that Graham brought from Jean-Louis saying, why are you doing design space? And they brought it up and they said, well, if you want flexibility and you declare it as a design space, you have completely different requirements than if you are not claiming this flexibility, right? And I think this is where the distillate of the whole thing is.
So I think it is the crux, Dora, and I'm really interested whether or not you think the applicant here, given their understanding and the degree of controls and statistical process controls they had, would they have needed to claim a design space, or would the PARs and controls have been equivalent? I almost wonder if we're at the level of these two things are the same.
We plan to do any feed-forward control, as I will explain in the next presentation once I get the time.
So still stealing Dora's time for the really last two questions?
I just want to answer to that. When I saw this slide, I just hoped that you were sleeping on. Because I knew this question would... I do not want to have now a discussion about PARs. Maybe this is something we have to come up on the second day. I just would like that before we have this discussion tomorrow, please go to the initial guidelines and have a look at the definitions.
Yes,
that's only, but no
done
answer,
some homework. Yes.
No answer. I'm co-chair now. I give the privilege of the chair. No answer. We can discuss this tomorrow. So please go and have a look at the definitions.